My name is Luana, and I'm the head of R&D and collaboration at QKine. I've been with the company since day one, my first show experience with growth factors and cytokine, and today I'm trying to tell you the story of QKine, what we do and how we do it. So first of all, QKine is a company based in Cambridge, UK, and is a spin-out of the University of Cambridge, a biochemistry department. And we produce high-quality growth factor and cytokine to support stem cells, organoids, and cultivated meat research. All our headquarters are in Cambridge, and this is the building actually where I work, where the company is based, and everything happens in this building, from designing of the growth factors, so R&D, the manufacturing, operation, shipment, everything is within this building. So this allows us to have absolute control of all the chain from start to end. We supply a cytokine growth factor to over 37 countries. We do through direct sales to customers, but also using distributors like REPROCELL, for example. And it's a company that's been founded by scientists, it's run by scientists. One of the main purpose of the company is to solve some of the challenges that scientists working with cell system face every day. One of the challenges is the reproducibility of the experiment. Of course, working with very complex system, like can be an organoid or differentiation of anything that involves cells, it gives a lot of variability and sometimes quite difficult to pinpoint where the problem lies. And one of the goal of QKine is to remove the growth factor and cytokine as a way of introduce variability. We do a lot of comparative data in-house, and we noticed that when reconstituting growth factor from other supplier, the concentration that we expected were sometimes very different of what it was supposed to be. So we started doing a little bit of a study comparing the percentage of recovery of our growth factors compared to growth factors from another suppliers. And as you can see from the graph here, the supplier B, that we leave it like that, the concentration, the variation in percentage of recoveries is very different. Sometimes it was less than 50%, sometimes was 60, sometimes was 100, sometimes 120. Whereas our recovery percentage of recovery is very steady, around 100. What does it mean? Our customer researcher in general, when buy growth factors and reconstitute the growth factor, they assume that the quantity including the vials is the quantity that is written in the label. So if you have 100 microgram of growth factors of protein in one vial and you add 100 microliter water, you expect you have a concentration 1 mg per ml, and all your dilution will start from that assumed concentration. If the concentration is actually 50% lower, that means that all your dilution starting point is wrong, all your dilution will be wrong. So this introduces variability into your experiment. So at QKine, we make sure that the concentration that is the quantity inside the vial is what we say it is. So we have very stringent quality control metrics, and everything has to be within a specific range, in particular case 95% to 115% of recovery. So the customer knows that once if they buy 100 microgram of protein, they actually receive 100 microgram of protein. And this reduces the variability also in their experiment. And also if you pay for 100 microgram, I think it's just fair that you actually receive 100 microgram and not half of it. We at QKine, we produce cytokine and growth factors completely animal-free. That means that we use a non-mammalian expression system. In particular, we use E. coli. And everything that comes into contact with the product from the very beginning to the end is an animal. It has to be animal-free. That means that any buffer, any salt, any plastic that we use during the process has to be certified as an animal-free component. So we require every supplier to give us certification of animal-free. So you are 100% sure that nothing that has touched an animal has then been in contact with the final product. We have been quite busy, and we're very proud to have released to the market few growth factors that nobody else's sells as animal-free proteins, in particular IGF, TGF-beta1, DKK1, activin E. That's a very important drug discovery target. And the last one that we released just yesterday is interleukin-34. It's the only tag-free, animal-free growth factors. It's very important for microglia differentiation. And we are really proud of that. There's a patent pending for this protein. It's been a long project, but really, really proud of that. Animal-free, this growth factor cytokine that we supply as animal-free allowed our scientists to have entire protocol completely animal-free. So with the release of IGF, for example, that meant the hepatocellular differentiation is now completely animal-free. So for our scientists and customers that want to differentiate iPSC to hepatocytes, they can do it using all animal-free growth factor. But why is it so important that we use animal-free growth factors? It's not just something about ethics or regulatory issues. It's more about but it's also about consistency of results. Using growth factors or cytokines that are expressed in mammalian system, either human or insect cells, can carry over some contaminant from endogenous expressed growth factors. This case, one of these cases has been actually published a few years ago. This particular paper is focused on GDF15. So this group was doing some experiment using a mammalian expressed GDF15, and they noticed that this protein used to was able to activate the SMAD2 pathway, but the activation on the pathway wasn't always constant, wasn't always reproducible. So some batches were able to activate this SMAD2 pathway, some other batches were not. So they started trying to dig out a little bit and understand why there was this variation. So they switched to an E. coli expressed GDF15, and they noticed there never was activation of SMAD2 with any batch of the E. coli expressed GDF15. And to cut a long story short, they realized that the activation of the SMAD2 signaling pathway from the mammalian expressed GDF15 was actually due to contamination of a TGF-beta1 protein. So TGF-beta1 and GDF15 are part of the same superfamily, so they are biochemically speaking very similar. So it's not unusual that you carry over a little bit of other protein that have the part of the same superfamily. TGF-beta1 is actually quite potent, so even a femtomolar contamination can activate the SMAD2 receptor pathway, signaling pathway. So switching to an animal-free protein will remove this risk from the beginning, because E. coli is not able to make any growth factor unless it's overexpressed from the operator. So this is moving from a mammalian expressed to an animal-free, so microbial expressed growth factor will remove the risk of target effect from contamination. Innovation is something that we love at QKine. So we try to understand what are the challenges of our researcher and try to think about a bit outside the box and try to release protein that can give a solution to some of the problems that our customers face. Now, 30% of our catalog is actually unique proteins. Those are proteins, they are a specific characteristic that can be either resistant to a particular negative fit loop, or like polystatin resistant activin A, or they have additional characteristic like FGF2 or a thermostable, which we create a thermostable variant. This is an example, and this is the FG3 FGF2. So the wild type protein is very unstable, high temperature. So if you incubate at 37 degrees for 1 to 2 days, it's already after 40 hours actually the activity is already decreased. That means that the researcher needs to change the media quite often. It's a waste of time and money, because you need to supplement the media with fresh FGF2 So we launched in the market FGF2G3. This is a thermostable protein. And here we did some experiment incubating the protein in media for 37 degrees. This is time 0, and you we compare the wild type that is in blue at the thermostable FGF2G3. And you can see the activity at day 0 is the same. If we leave it for 48 hours and we again check the activity, we see that the wild type in blue has already lost significantly activity, while the FGF2G3 is still very much active. We also collaborated with University of Turin in Italy with Professor Bertero, and Professor Bertero tried a couple of FGF2G3. We have 2 different lengths, a short length and a long one. So the 5.4 and 5.4 and 4.5, and he did some experiment trying to see if there was some difference in the length, and he found out actually the shorter FGF2G3 in his system, it was actually more potent than the longer version. Also, of course, BFG3 more thermostable. So he managed to reduce the spending of media from 650 euro per liter to 80. It's because he needed to use less because it was more potent, but also there was no need for changing media every other day, so reduce cost there and waste of time as well. And this is again more extensive comparison of thermostability for the FGF2, comparing the wild type day 0, day 2, and day 7. So after a week, it's pretty much no activity detected. With the G3, this in particular, the 145, the Alessandro Bertero tried. After 7 days, still pretty much the same activity. So you can use, you don't need to replace every other day. So it's a weekend-free protocol. We have a 9-point quality commitment statement at QKine. So we make sure that all our growth factors are animal-free and there is full traceability. We operate in an ISO 9001 manufacturing facility. We have an accurate protein recovery from vials, as I explained before. Bioactivity is reproducible, and there is no lot-to-lot variation, as I will show in a moment. And we have the highest purity level in the market, so the iron 98% purity. Low endotoxin level, so most of the time are under detectable level from testing, and we have sterility and mycoplasma testing as well. This is an example of lot-to-lot consistency. So it's particular proteins, BMP4 is also a member of the TGF beta family, which at QKine we are expert of. And in this graph, you can see 4 different lots plotted, testing the activity, and you can see there's pretty much every lot is the very similar activity. And one thing that we do when we you see our batch, the activity needs to be compared with the batch before. So we ensure that the activity hasn't changed, and regardless we lot our customer by the end result will be the same. As I said, we do a lot of comparative data and analysis, and this is a comparison of our BMP4 QK038 with a mammalian express supply A and E. coli express supply B. Our results be more potent, both A and B. This is due to the fact that it's much purer and higher quality. Apart from the research use only grade, we supply also food grade protein. Those are for specifically for companies that work within the cultivated meat market, and we supply species specific growth factors to help with the FDA recommendation to remove human protein for production of cultivated meat. So we have bovine, porcine, salmon, eel, tuna, and whatever flavor that you think about. We have it, and if we don't have it, we're probably happy to make it for you. This range of protein is go through the same QC that the research usually goes through, plus some additional QC and particular DNA and protein or cell testing to make sure that there is no trace of DNA or protein coming from E. coli. We supply with toxin and allergens certification for if required, and we help with any regulatory report if needed be as well. Coming soon, we have a cell therapy grade. So those the growth factor within this range will be manufactured to an ISO 23999 standards in an ISO 14644 aligned clean room. And this grade will be released in a month or so. So stay tuned. So if you are looking for a cell therapy grade growth factor with high quality QKine supply, please get in touch. And that's me for today. Thank you very much for your attention. And if you have any question, please don't hesitate to ask.